Good morning. Welcome to Semel Grand Rounds. We're going to have our Child Division Chair, Dr. Suma Jacob, introduce our speaker for today.
Thank you, Sabrina. Good morning, everyone. I'm very excited to introduce Jeremy Veenstra-Vanderweele, who is here for the Denny Cantwell Lecture. And he's a Ruane professor at Columbia and the Child Division head there. Jeremy has so many different accomplishments that it's hard to summarize. So since this is the Denny Cantwell Memorial lecture, I thought about some descriptions of Denny that people like Kathy and Jim McGuff would remember, and thought about how Jeremy's similar to those. And one of those is leading educator and researcher in broad range of groundbreaking studies. So in terms of research, Jeremy does work that spans animals to humans in autism and OCD. And he's published over almost 200 publications, and is truly, some of the work is groundbreaking because there's reverse translational studies looking at restrictive repetitive behaviors within autism and overlap with OCD. And another characteristic of Denny was he was beloved by students and professionals for his incomparable teaching. And Jeremy is really active in teaching. He teaches the Institute at ACAP often. I often go to ask him questions related to teaching. And one of my favorite stories about his dedication to education is, as many of you know, Columbia's had some challenges, and he worked really hard to find funding to keep trainees and junior faculty afloat during these challenges. So he's really committed to the next generation in the field. Denny grew up in East St. Louis, and I love that Fighting Irish picture in the library. Jeremy and I, although we didn't know each other as kids, grew up in Southside Chicago, which is known for the Fighting Irish, and had St. Patrick's Day this week. And so we totally get that culture, and in some ways love it, even though neither of us are Irish. Denny trained with Michael Rudder, and so did Kathy. And Jeremy, who I knew at the University of Chicago, trained with Kathy and Ed Cook, and these incredible people who are also, well known in the world for autism. And then Denny was the recipient of many awards, and it was prolific with publications like Jeremy. Jeremy is a recipient of many awards, including the George Tarjan Award for Contributions in Developmental Disabilities from ACAP. So I'm going to have him share new science and knowledge and updates.
Thank you, Suma. And I realize, okay, so now I understand why you were standing back a little bit. So I guess you can't point there, you can't point, we'll figure it out. I will say it's really a pleasure to be here. I got to give a CART talk now, I don't know, before COVID, which means what, a decade ago? Which was really fun. But when Suma asked if I could do the Cantwell lecture, I was both touched and intimidated. When I was coming up as an undergrad, but certainly a medical student and then a resident, Denny Cantwell was part of this sort of pantheon of godlike figures in child and adolescent psychiatry. Many of them died too soon. And so I heard about Denny from folks who trained with him, including Brian King, Jim McCracken, certainly folks in this room, Kathy and others. So it is really, it's an honor to be here and to get to visit with you in Denny's name. And certainly will not try to live up to what I read over the last few hours to understand again the impact that he made. I will just note that I'm going to talk about some medications in development. None of them have made it to the market. And when I was a child psychiatry resident, actually when I was an adult psychiatry resident, the first year I ate all the drug company food and was like, this is amazing. And the second year was, this is just wrong. And figured out once I was a faculty member that it's not possible to bring medications to market without engagement with industry. I'd be happy to have that as a discussion. None of this has made it to market, and so the conflicts will show up in red when they're relevant, but by and large, they're not relevant to clinical practice yet. And then we'll note that this is sort of a conceptual talk, so I'm not going to talk about some of my own restricted interests, like the serotonin system in autism. Oh well, so if you came for that, you can leave now. But I'm going to talk conceptually and won't therefore have a chance to highlight a lot of the folks who've done some of the work, whether in my group or elsewhere. And then really the talk is just disclosures, and then we'll go and eat lunch. But I just will note that at this point, if you are coming expecting to hear that we have interventions that are based on in biology and autism, we don't. I'm sorry to ruin that. And so instead, we're going to talk about pathways and how we might get there. And I should just acknowledge in all humility, I don't know. None of us really know. And so I'm going to outline how people are trying to get there. And then I think this is the last disclosure side. We'll acknowledge that terminology is still something that we're wrestling with. Even, you can see I took spectrum disorder out of the title of the talk, so it wrapped too long on the slide. But as we talk about people with a diagnosis of autism, we use a variety of different language. And so you'll hear me go back and forth between describing autistic people, usually talking about people who've identified themselves that way, and talking about people with a diagnosis of autism, thinking about the medical model. These two ways of thinking, medical model versus identity or neurodiversity model, oftentimes don't work well within the same sentences or paragraphs. And I just want to acknowledge that and approach with humility, and happy to have that discussion afterward as well. I think the primary thing is sort of thinking about what we're hearing and reflecting back to people the language that they prefer. So this is what I'm going to try to walk through. And we'll just acknowledge, I'll start very briefly on a couple of sort of conceptual things that are going to be very familiar to folks in this audience. So when we think about autism, we're simply not discussing something that is unitary. And you hopefully understand that. The definition has changed over time. A lot of that is due to folks like Kathy, who acknowledge we can see differently once we really see with rigor. But when we see somebody with a diagnosis of autism, we're usually focusing in the center here on what leads them to meet diagnostic criteria. And certainly when I see somebody at the Center for Autism and the Developing Brain, Kathy started, they essentially have to meet those criteria in order to walk in the door, at least to get to the point where they see me. But oftentimes I'm not seeing them for their autism diagnosis. I'm seeing them for another reason. And you could put that in the middle just as easily. And so we have to acknowledge this is a talk about potential interventions in autism, not necessarily for autism. And when we talk about interventions in autism, we could talk about somebody who sort of fits this pattern, where they have language difficulty, where they perhaps have a seizure disorder, somebody who fits this pattern, very different, right? Other things around the sides. And in both cases, we're talking about potential interventions in autism. And we just have to acknowledge that that probably is not the conceptualization we will have in 10 years, and certainly not in 100 years. I'll also note that back and forth, and when we first started to use the term autism spectrum disorder, some people would say disorder singular, and some people would put an S on it. People like Dan Geschlin and Pat Levitt published papers with an S on it. And that is in part based on this sort of model where, really we're describing people who have different discrete identities, different colored balls in a ball pit. And once we know what those are, we'll be able to say, okay, well, you're the blue kind of autism and you're the yellow kind of autism. And I don't actually think that's likely to represent things very well either. We are likely to have molecularly based diagnoses that might fit that pattern. But when we see somebody in the clinic, we think more like this, right? An individual who has areas of strengths and weaknesses, and some of those strengths and weaknesses are enough to meet diagnostic criteria. When we think about risk, and this is a slide from Ed Cook, one of the folks I had the privilege of hearing about Danny Cantwell from, we still have a lot of data that favor a model where multiple risk factors coincide in an individual to lead to a likely diagnosis of autism or something near autism. And yet most of what we actually can hang our hats on are these relatively rare things that contribute most of the risk for an individual. And that doesn't integrate particularly well with this model at all. I'm going to use Fragile X syndrome as an example of that in this talk, but there are quite a lot of examples that could be used. Some of these have started to, oh, and I should say, on occasion, you can also point to an environmental risk factor that fits this pattern, like exposure to valproic acid in utero. Thalidomide exposure is another one. We may also find others of those over time, but in those instances too, you can say, if they didn't have exposure to valproic acid in utero, if they didn't have Fragile X syndrome, they almost certainly wouldn't have autism. So it's not directly causal, but you can do the causation in reverse. Some of these overlap a bit from a molecular perspective. So folks here have studied some of these, folks elsewhere as well, where you can say this same molecular pathway is involved in multiple of these syndromal disorders. That's actually not usually that clean. And in fact, some of the work from Dan Geschlin and others points to pathways, but the pathways sometimes are perturbed in one direction versus the other, and we don't yet know how that's going to translate to intervention. And there are prettier ways to represent this, but I'd like to keep it just conceptual. This cascade from what we understand about risk to what we understand about behavior has a lot of way stations in between, and we don't yet really have a clean path to understand all of that pathophysiology. But we do have an important perspective that the roles of genes, proteins, systems are different across development, and are starting to incorporate that in our study designs. But what we can, I think, count on is that there should be some level of convergence as you get closer to the top of this inverted pyramid, where we're identifying individuals who fit diagnostic criteria based on behavior. And you would expect as you go back down that cascade that you should have some things in common across individuals. We'd like to think that that will lead us to biology-based interventions. But I will say at this point, most of the convergence that we can point to is at the level of protein networks or cells to a lesser extent, although increasingly, with a lot of uncertainty in between. That's still a lot better than our previous model. And I will say our previous model is pretty great in some ways, right? Early intervention, we know makes a difference for a lot of kids, but you don't need to know a thing about the brain in order to implement what we do right now, unfortunately. And that's true on the behavioral side and the medication side. I'm going to very briefly talk about those pieces, what we know, and I'll say I'm going to spend very little time, sort of the least time, on behavioral interventions in this talk because we're talking about biology-based interventions, but that's where most kids with autism should be spending most of their time. And we know that early intensive developmental and behavioral interventions make a substantial difference for a lot of kids. We also know that there are more targeted interventions that can make a substantial difference as well. And there's accumulating evidence. I will say, and this is a challenge, if we hold ourselves to the standards that the Food and Drug Administration has used over time, most of these interventions would actually, at this point, still not meet the criteria for FDA approval. We think that they work. We think that we should be doing them, but there remains some uncertainty. And to just share a brief example, and there's a follow-up paper I'm not going to show, but when I say substantial impact, we're talking about a standard deviation of IQ movement over the course of intervention. And yes, you can see movement with community-based control as well, but that's not a small degree of impact. It's something we should hope for all the kids that we see. On the medication front, I think it's instructive and disappointing to think about sort of the top two medications that have had the most randomized controlled trials in autism. So oxytocin people usually recognize as the one that's had the most trials, although most are very small. Anyone know what is the second in that line? Kathy probably knows.
It's the pig hormone.
So it's yeah, secretin. Yeah, absolutely. So secretin, and I use this as an example because no one still believes that secretin is effective. But going back now 25 years, secretin was first reported in a case series of kids who improved after a GI procedure. and was reported to lead to dramatic improvement. So it was featured on things like 2020 as something that, you know, really was transformative in autism. Lots of kids received injections of secretin. And I will just say, this is one of the larger studies that looked at secretin versus placebo. You can see secretin led to substantial improvement when measured with a parent report of developmental symptoms, but placebo, actually, that's what you're seeking, right? So placebo is even better. And I think it's just notable that when you're using developmental outcomes as your outcome measures of choice, kids continue to develop. And so if you say, well, I'm going to implement this treatment, is your child developing? The answer is almost always yes. And so when we're quantifying that, it becomes very difficult to separate from placebo. Unfortunately, because we don't have a lot to offer outside of the behavioral realm, a lot of families remain excited about interventions that have pretty limited evidence. And this is, I mean, this is shocking, right? 69% of families, after being shown the data, still felt like secretin was worth continuing to consider. All the medicines we have right now treat behavioral outcomes that are not core symptoms. And so risperidone and aripiprazole, I'm not going to spend time on. These are FDA-approved medications. I will say here too, you do see some placebo response in these studies, but you see a clear separation and advantage for aripiprazole, multiple different doses of medicine, and you see better improvement when you actually allow the clinician to adjust the dose of medicine rather than sticking to a set dose. We also can treat ADHD and autism spectrum disorder, and I will say from a clinical perspective, this is something that I thought was really, really boring when I came out of child and adolescent psychiatry fellowship. Treating ADHD and autism, you know, okay, we all know how to do that. There's an art to it, and importantly, if you improve kids' ability to pay attention, you improve things like joint attention. And so there's a real point to treating ADHD and other things that get in the way of kids learning. I call this the 50% rule. Essentially, you pick any of your common ADHD medicines, and you have about a 50% chance of that being helpful in autism. There's a little more art to it than that, but you do expect to walk through different options to find something that's most helpful. Okay, that's all I'm going to say about what we currently know, and then I'm going to talk about the pathways that may lead us to new knowledge. So I'm going to start with the example of rare genetic conditions and how that may lead us to biology-based interventions that really are intended to go up that whole inverted pyramid. So there's lots of different things that you can think about, and we've done work in Fragile X syndrome. I pulled out a study design that is in 16p11.2 deletion syndrome. I will say that there are lots of other things that we could be talking about. There's ongoing work that I think is going to be exciting, but I'm going to use Fragile X because that's where a lot of this has been pioneered. So Fragile X syndrome is one of the more common genetic conditions that you identify in autism, but I will note that usually today a kid comes in with the diagnosis of Fragile X syndrome instead of getting the diagnosis after the autism assessment. And so most of the time, if you see a child with an autism diagnosis, you're not imagining that you're going to find Fragile X syndrome. I have, however, diagnosed Fragile X syndrome in a 30-year-old in my autism clinic who was there for behavioral concerns and had never been picked up, in part because we all imagine that we can pick these kids out of a lineup because they look a certain way. But the way that they look, the prominent ears, prominent forehead, prominent jaw, blends right in my family, as Semel could tell you. And so they do typically look different from family members, but they don't necessarily look different from every kid out there. And we just have to be aware that we should test instead of making the assumption that we'll pick up on appearance. These kids have mild to moderate intellectual disability, usually moderate intellectual disability. You can also find Fragile X syndrome in girls, although many fewer are diagnosed, in which case you see milder intellectual disability. You see autism, meaning meeting full criteria for the current definition of autism spectrum disorder about a third of the time. A larger number have social difficulties. They come across as bashful more of the time. But if you didn't know they had Fragile X syndrome and you just had 100 kids with an autism diagnosis, you wouldn't say based on behavior, oh, this child has Fragile X. That makes sense. They tend to be hyperactive and impulsive. that tends to fade some over time, just like ADHD usually gets better in that domain. And they can have seizure disorder, but most don't. We've known for a long time, now going back about 30 years, that there's an expanding trinucleotide repeat in the 5' untranslated region. If it expands past 200 repeats, you get hypermethylation and silencing of the gene and lack of the gene product called FMRP for Fragile X mental retardation protein. This has a variety of roles, but serves as an RNA chaperone. And I will say one of the roles that it plays has drawn a lot of interest and excitement over time. And that role is within glutamatergic synapses. You see here, and forgive me for those who like to look at actual pictures of synapses or neurons, but presynaptic neuron releasing glutamate, we're usually thinking about action on AMPA and MDA receptors, but a metabotropic glutamate receptor, mGluR5, nearby actually serves to dampen some of the downstream signaling. And Fragile X protein is actually translated in response to mGluR5 signaling and then serves as part of that dampening process. So if you don't have Fragile X protein, you see too little AMPA receptor in the recirculating pool, so too little on the cell surface, and you see synapses that fail to mature appropriately. You can see that under a microscope because you're then not suppressing nearby dendritic spines that are reaching out, trying to make more appropriate connections. And remarkably, and this is one of those amazing examples of translation across species, I could be showing you the neuropathology from human or looking at the mouse equivalent under a microscope, and they're essentially indistinguishable, where you see these long filopodia-like dendritic spines reaching out in the Fragile X model, and you see more mature appearance in the wild type or the typically developing human. So that's a beautiful parallel. From a behavioral perspective, the mouse is a little bit hyperactive. It shows deficits in certain social tests that don't easily translate. They will more likely back out of a tube when confronted with another mouse, and some impairments in learning that also don't easily translate. So they fail to learn appropriately how to avoid shock in certain contexts. But remarkably, because you can see it under a microscope, you can then test different ways to potentially intervene. And this is an example where a group from Roche, although this has been done by other teams, used a negative allosteric modulator, a medicine that dials down response at that receptor, and was able to show that when you give that shortly after mice are weaned, extending all the way to sexual maturity, a lot shorter in a mouse, that you can rescue the appearance under a microscope, that you can rescue all the behavior that you can point out as different, and that importantly, that potential medicine has no obvious ill effects on health. So really remarkable, exactly the sort of thing that we'd like to translate. And then they took this idea that, okay, we have some data going all the way up to the synaptic level. We have some idea of behavior, at least that's different in Fragile X syndrome, and tried to jump, although acknowledging that jump into the human doesn't really have clear parallels from a behavioral perspective. And in that jump, and both Roche and Novartis did this in parallel, they had to start first with adults for regulatory reasons. The Food and Drug Administration insists at least that you have safety data in adults before you move down. And I realize this is hard to see, hopefully easier to see on Zoom. The gray is placebo, and you see actually that if anything, you want to be on placebo again instead of active medicine. The outcome that they were looking at was aberrant behavior, sort of lumped in across a scale that's oftentimes used in individuals with autism or developmental disabilities. In adolescents, also, at least in the condition that was expected to see benefit, the placebo was, if anything, better. And so this led to a lot of discouragement and some loss of jobs as both Novartis and Roche closed their programs in Fragile X syndrome because it was viewed as too big of a risk to try to keep carrying on. There are lots of reasons that these trials might have failed. I will say a lot of us thought it was because they were looking at the wrong age. You would never say, well, this is a developmental disorder that we can identify before a child is 3. We're going to start in 35-year-olds. So a lot of us thought that, some thought wrong outcome measures, and then there are ongoing questions about how well you can really expect this particular protein to be translatable between mice and humans. So we were part of a study that followed this up, that now at least has had the initial data published. And the idea here was to look earlier in development, so 3 to 6 year olds instead of 36-year-olds, and to try to dial down. And I actually can't tell you, there was a lot of difficulty getting the EEG measures and eye tracking measures in this population. So I can't tell you whether that's going to end up yielding anything yet, but tried to dial down a little bit further than simple behavioral outcomes. The outcome of interest was language learning. And these kids were provided an enriched intervention, a language enrichment intervention delivered by their parents with coaching from a speech and language pathologist. I'll say again, this is probably difficult to see from the back. The blue line is placebo, the red line is active drug. And if you can't tell the difference, it's because there was really no difference. If anything, again, the placebo may have been a little bit better. You can see, unfortunately, there's very little movement on this scale over the course of the study, which probably means it's not an ideal outcome measure. But you can also say it probably is the case that this is something that doesn't move language learning in the way that you might have hoped. So that's depressing, and I will acknowledge that this talk may be depressing, so my apologies for that. But I will say there are lots of other ideas of how we might be able to intervene. There are other medications that are currently reaching the late phase 2, early phase 3 trials where you're really going to be able to see whether there's an advantage. And some of those may be helpful using the methods that have been developed to test this hypothesis. And this cascade, I will say, is a hopeful cascade. Although there's a lot of uncertainty about how we get from where we have mechanism identified, at least one potential mechanism in animal models, to where we have treatment studies that actually show a difference. And I think there's a big question there. My brother is an oncologist, and so he's experienced this amazing surge in new medicines over the last decade or so, which is, I think, phenomenal. And I will say, until the early 2000s, late 90s, early 2000s, the oncology cascade didn't look that different from this. But there's been a huge advance once they developed the methods, once they had the targets available. I don't know that we can expect the same in developmental disorders, but I do think that we are likely to see a tipping point where we actually have the methods available to identify differences. I will say there's also a way to short circuit this, and I'm going to talk about that just very briefly, which is potential genetic interventions that go directly from identifying a genetic risk factor that contributes much of the risk to potentially implementing genetic-based treatments. And that jump is illustrated by spinal muscular atrophy that we're all now pretty aware of as an intervention that uses, yes, a spinal infusion, so that's pretty invasive, but actually leads to a transformational change in children's motor outcomes, but also we expect their lifespan. And that's based on a pretty simple intervention where they're actually using an antisense oligonucleotide to activate a pseudogene, a gene that would not ordinarily be able to produce a useful protein, turn that on and allow kids to have that gap in protein signaling be filled. And to have really transformative outcomes. So I have a colleague who's a pediatric neurologist who spends a half day a week doing this and feels like that feeds her for the whole rest of the week, where she's seeing a lot of kids where interventions aren't possible yet. So this is likely to be at least part of the future, and I think there are big questions. Right now, we do genetic testing after autism diagnosis, at least some of the time, not for that many kids actually, but we can expect in whether it's 5 years, more likely 10 to 15 years, that we will have, instead of a newborn screen that's simply metabolic using a blood spot, we could use the same sample and do newborn screening with sequencing. As sequencing cost comes down, that is more and more practical. In a study called Guardian, led by Wendy Chung, with the initial paper recently out, the Columbia New York Presbyterian Hospital System has been doing just that, actually using whole genome sequencing on the newborn spot at birth, and then delivering the results back to families. There are two different sorts of results that can be returned. those that are immediately actionable, that are essentially the current newborn metabolic screening plus another 100 or so conditions that we think are actionable, and then a level 2 screen that would include returning some neurodevelopmental results, particularly when there's risk of epilepsy or where there's enough developmental risk that we expect that early intervention is going to be indicated. And we're part of a study, although I will just say that the NIH canceled our funding last Monday. We're still going to do at least parts of this, where we're taking the kids who are identified at genetic risk and following their neurodevelopmental trajectories over time. That's work done by Dina Anzo and Bill Pfeiffer. Doing a later assessment of diagnosis that's led by Steve Canny, and then also doing interviews with the parents as well as a lot of surveys led by Paul Applebaum to understand how families absorb this information, with the idea that we currently can identify the risk early, but I can't tell you that we have enough confidence to say in which kids you'd consider a genetic intervention. And so we really need to be able to say, well, in which cases would you consider that? In which cases would you not? And also down the line to be able to say, in which kids should you begin developmental intervention immediately? And in which kids do you wait until they actually have an identified developmental delay? So I'm then going to pivot and talk about sort of top-down types of interventions. And this is most of what we do in psychiatry right now, right? We don't have precision medicine. We have what a family on Monday, well, I guess it's yesterday, described to me as, yeah, you know, we've been through a lot of trial and error with our psychiatrists. We're not sure they're any good. That's just what it's like to see a psychiatrist. So sorry, I said that in psychiatry grand rounds, but this is what we have to offer. And when we approach it with humility at the front end, we can help a family understand we're going through the evidence base and it is going to feel like trial and error, but we're really doing things in a way that follows the evidence. And what I do oftentimes starts with my favorite question in the early assessment, which is, are they constipated? And if they are, I can do something about that. You want to make sure that you're identifying epilepsy, and that's something that you know will improve their developmental outcomes without ever doing a randomized trial to say that it does, right? This is sort of like, you know, you don't do an RCT of kindergarten, you don't do an RCT of parachutes. We know a lot of our interventions can improve language outcomes. Oddly enough, IQ is still probably the most robust outcome for most of the early intervention literature. So we know how to, even though these aren't core symptoms, how to impact these areas. And most of what I do as a child and adolescent psychiatrist fits in one of these realms, which clearly is not core symptoms, but can make a big difference. And I will say, we also know that there are components that can contribute to an autism diagnosis that are not necessary to achieve a diagnosis. So some kids are very sociable, very interested in others, and just struggle to navigate that interaction. And other kids would really benefit from an intervention that led them to be more interested in other people. And so I'm going to posit that if we had interventions that were more symptomatic, so not targeting the full core symptoms, but potentially could act on things like sociability, or maybe could act on a subdomain within repetitive behavior, that these might actually benefit a larger group of individuals, but are unlikely to be truly transformative. They're certainly not going to be something that leads to a loss of a diagnosis. But this fits sort of up here, right? And I think we're all hoping that we have this sort of a convergence that we can target. And I'm going to use oxytocin as an example here. I will say, I'm going to do this very briefly. I still think the oxytocin system is very interesting, including in relation to autism. I will just say that what we've done thus far, and I'll give you an example of that, hasn't really harnessed that potential impact. So we're all quite familiar with oxytocin for its roles in a number of things, including reproduction, including sexual behavior. In autism, the findings are quite mixed. So there's some data potentially pointing to oxytocin as a biomarker, but I will say there's differences in terms of the age where you look, and considerable question method to method. And Suma could explain this way better than I could. And there's really quite mixed genetic data pointing to the oxytocin receptor, not anything that you would say is consistent. But oxytocin can actually move social behavior in humans, or at least in groups of humans. And this is an example in autism, although there are many examples in the general human population. There's a French group that either administered intranasal oxytocin or a placebo comparator, either to folks who are defined as healthy or individuals who had an autism diagnosis who are described as quote unquote high functioning, which means that their language function was intact at minimum. And you can see if they received a placebo, they didn't see any difference, whereas if they received oxytocin, they did on this cyberball game, where the individual thinks that they're playing catch with somebody who's in the other room. They either can play catch with an individual who throws the ball back or somebody who doesn't. And you see that when you receive oxytocin nasal spray, you're more likely to play catch with the individual who throws the ball back. Not something that's part of the ADOS, but shows that you can move social behavior. And there are other examples, similarly in a laboratory setting, including other examples in this study where that was the case. In this study from Kevin Pelfrey's group at Yale, they tried to extend this into the scanner. They actually did not see behavioral differences with intranasal oxytocin, but when they compared response to eyes, social stimulus versus vehicles, they saw that there was a differential response with those who received oxytocin versus placebo. And that included areas like the nucleus accumbens, so maybe social reward, and other areas that they related to the social brain. Importantly, they did not see differences in behavior. So you can move the brain or behavior or maybe both. Following that, and actually in part before that, a number of groups did sort of pilot level studies of intranasal oxytocin. This is one of the ones that drew the most attention, where there was an advantage of oxytocin over placebo on the SRS that was only statistically significant once you controlled for baseline oxytocin levels. And they actually showed changes in oxytocin levels in the placebo group that are a little difficult to understand until you realize that oxytocin may be part of the mechanism for placebo response. So oxytocin now has been squirted up lots and lots of noses. I'm not sure that's what we want to do, but usually it's available for free shipping. And we were part of a study not to ask whether the oxytocin system is involved or can be manipulated in autism, but to ask whether what most people were doing at that point was actually likely to help. We had a large number of participants. We were able to slice the population in different ways. And I should just put up the same graph and tell you it's a different. And we saw no advantage for active medicine versus placebo across nearly 6 months of treatment. So very disappointing. I will say we ramped up the dose of oxytocin over the course of the trial, but it was really designed to mimic what was being done out in the community. It was not accompanied by social skills training. It was not in a particular population that had low oxytocin levels, although we tried to look at that. And we were unable to do some of the really fine-grained things that you'd like to do to say, well, does the cyberball game move, even if their real life behavior doesn't? Do they show changes in eye tracking? We couldn't do any of that, but we could say that the real world behavior didn't clearly change. So I'm going to shift to talking about sort of where we might be able to refine things moving forward. And I'll give one example. It could be a whole nother talk about sort of other potential examples, but this is an example of what I think we're hoping for, that we find something that fills the gap between this very narrow precision medicine approach and the broad symptomatic treatment approach. And I'm going to use the example of a GABA agonist that has been tested and not fully ruled out yet as a potential treatment in autism, although I will say I wouldn't recommend this as the next step for a child you're seeing in the clinic. So the idea of using medicines to manipulate the GABA system came from a number of observations, including that epilepsy is more common in autism. There's some data from mice, some data from other species suggesting that if you manipulate the GABA system, potentiate the GABA system in some models, you can see changes in social behavior. I will say that there's also some evidence pointing the opposite direction, so I don't want to make too much of that. But we were part of a study that used a GABA-B agonist, arbaclofen, not available for general use. And in a general population of individuals with an autism diagnosis, the primary outcome measure showed no benefit whatsoever. That was a measure of social withdrawal. Yet what was seen was that some kids actually showed an improvement as rated by their clinician in an overall measure of severity, the clinical global impression. And to just note, this is the primary outcome measure. Again, very difficult to say that that's two different lines. And then when we look at this measure of severity, you can see that there's separation that starts already maybe by 4 weeks and extends certainly to grow by 12 weeks. But when you look at the individual ratings, and this is a very simple 7-point Likert scale, that's sort of an overall rating of severity. The difference really is seen in the placebo group, where there are fewer kids in the active group that show no change on drug versus placebo. And that's mostly because there are a few kids over here, it's about 15% of the total, who show pretty sizable changes in clinician ratings of their overall severity. I will say we had one of those kids at our site, and I don't know whether this was really due to the medication or not, but this is a kid who shifted from not using language functionally to using language functionally. So these were not minor changes as described by the clinicians, but it's not enough to hang your hat on. When you looked at the subgroup analysis, you saw most of the big changes were in younger kids, kids who had milder symptoms, and kids who had higher IQs. So this is not something that I would hang my hat on at this point. We need to understand who these kids are who are actually showing this improvement, if that's something that replicates. And there now is data, some of it's been presented from 2 studies, but they're being analyzed as one study, suggesting that there's a hint of signal in a larger population, but certainly not conclusive. And I mentioned this study that we're part of in 16p11.2 deletion syndrome that is using arbaclofen with the idea that the animal model and some data from humans suggests that there may be some more specific effects in that syndrome. What we'd really like is to be able to sort of fill that gap, to identify, well, maybe it's kids who actually have evidence of an imbalance between their excitation and inhibition in a particular part of the brain. We'd like a biomarker to be readily measured, replicable, and robust. I think there's a lot of questions about what it might be beyond that. Does it need to be tied to genetics? Do you need a clear line where you can say binary, this group has the biomarker, that group doesn't? Could it change with treatment? Do we know why the biomarker is there? And I know that UCLA has been part of the ABCCT over time that is trying to focus on a particular biomarker that relates to social response, where we don't have any idea of the potential pathophysiology, but it might still be useful as an outcome. And we'd like that to fit between this precision domain, where there's a big impact of treatment, but a tiny number, and symptomatic treatment, where there's a small impact, but a big number. I don't know exactly where the sweet spot is there, but I will note that since we started with Fragile X syndrome, there's a whole network of genes that are bound by the Fragile X protein, or whose mRNA is bound by the Fragile X protein. So that's one way you could think about things. Or Dan Geschwin's group just published a study in postmortem tissue showing networks of genes that appear to be co-regulated and related to one another. It could be that that would offer you an opportunity for a genetic basis of a subgroup identified by a biomarker. It could be neuroimaging. So I mentioned the finding from Jamie McPartland's group and others of an EEG response. Okay, is the echo still there? No. Of an EEG response to social stimuli. It could be something as crude as just whole body growth. So there's this subgroup of individuals that Kasia Szwarska and plenty of others have described that doesn't just have enlarged heads, and that's certainly seen in a subgroup of individuals with autism, but overall enlarged bodies in relation to their family members. And it may be as much as 15%. We know some genetic syndromes that include whole body overgrowth, kids that are bigger than their family members. That seems like it could be a biomarker. So there are other examples that we could think about. But I do think that that is in this domain as participant selection. I will say down below that, you see outcome measures grayed out. That's a different talk and probably better given by people like Kathy. But I do think that we would like to have outcome measures that are better than just parent ratings or even teacher ratings of aberrant behaviors, circled on a bubble chart, and get to something that is more observable. And ideally something that can be replicated clearly from direct observation of the individual. And we're not there yet, in part, I would say, because we don't have the interventions that work that we would use then to validate the outcome measures. ultimately, I don't think that we should expect that biologically based interventions by themselves are going to lead to the sorts of improvements that I think most families are hoping for when their child receives a diagnosis, whether it's 18 months or 4 years. And I think that where we need to eventually get to is a combination of different intervention approaches. And this is one example, and I wouldn't say that this is the ideal example, but this is an example from the RUP group led by Mike Amon, where there was both a medication intervention, in this case risperidone, plus a behavioral intervention with a modified version of PCIT, quite modified. Most people describe as Ruby. And you can see here that these groups do separate over time by the end of intervention, not by that much. And at 6 months, there was no longer a significant separation between the two. Importantly, the medication group here shown in pink ended up taking a larger dose of risperidone than the group that was receiving medication plus behavioral intervention. And that shows one of the challenges in trying to separate. You really need to hold one of the things steady and probably need to use a suboptimal dose of one in order to show an advantage of both together. And we will need to learn over time how to test things, because what we'd like to do is have a biologically based intervention that puts the brain in a state better able to learn, and then a behavioral intervention that actually takes advantage of that. That might be more like D-cycloserine, which is an example from the anxiety disorders and OCD literature. And I will say this is not as robust as it might be, but if D-cycloserine was still on patent, I bet it would be more robust. It's very difficult to do this sort of work using NIH funding alone. So D-cycloserine actually is a modulator of NMDA receptors, serves as a partial agonist. And you can see if it's given between 30 minutes and 2 hours before, and I will say that variance is because of differences between mouse, rat, and human literature, before an extinction-based CBT session, you see a more rapid rate of improvement in OCD and a variety of phobias, in some anxiety disorders that have been looked at, and some data in PTSD as well. I will say most of us don't use D-cycloserine currently, which is also an interesting thing. I would say part of that's probably about marketing. It doesn't have an indication for this purpose. At times, it's been hard to get your hands on. I have used it and have seen that kids I'm trying to help do seem to show a sort of an earlier breaking point on CBT where they see improvement. So I did leave time for questions, assuming that clock in the back is accurate. But I will just note again, I think we have to be humble that when we apply a diagnosis, we're applying what we know right now to an unknown sea, whether that's a ball pit or whether that's a mountain range. And we should acknowledge that and acknowledge that we need to do that in order to pick potential interventions, because that's how we study things, right? That our current treatments are not based on neurobiology, but can be very effective, and that we have, in some ways, these two quite different approaches to identifying new interventions. I do think precision medicines are going to be transformative in some cases. I don't think it's going to be the majority of cases, at least not in the near term. And these sort of symptom-based or circuit-based interventions, I think, are likely to also be helpful. And that's where we have examples. And we know that we can see improvements when we treat ADHD in a kid with autism. If we had more examples like that, I think it would also be powerful. And ultimately, we need to identify better biomarkers in this sort of joint approach to treatment. So these are some of the individuals involved in some of this work, and then folks involved in funding what I actually shared today. And then I can take questions.
Yeah, so your comment about large heads.
I also have a large head.
Well, no, I just want to start with that. So I saw a TikTok yesterday saying that
I'm in LA now.
that someone was proposing that having large head was a sign of being Irish, which I think Dr. Campbell would have appreciated. But more to your point of precision medicine, it looked to me from your list of potential gene targets, you're accounting for maybe 4% of AFD. I mean, just a rough maybe 5%. So I guess question one is how, at this point, what would the expectation be of how much? It'd be wonderful, like some of our metabolic things, you identify it at birth, and you
Yeah.
can give them a different diet. But how much impact would that have based on what we know now? And then the second thing related to that is, I think I saw another TikTok, how I spend my retirement. Chris Hemsworth has retired from is taking a pause from his acting because he has the ApoE gene, saying he's at some risk for Alzheimer's. He's got the money to do that, but what impact on a family to tell them that we've identified this mutation that creates some risk? To me, that intervention itself is going to shift what's going to happen.
Yeah.
Possibly. So how do you manage all those things?
So, good questions. I'm wondering, before you retired, did you talk at Grand Rounds about TikTok, or was that?
No, but I'm an expert right now on people alleging all bad things are because of their use of Snap, so we can talk about that at dinner.
So can people online hear the questions, or should I repeat the question? Okay, so I think the first part of the question is how much of the population might fit into this precision medicine domain. So currently, if you send both a chromosomal microarray and chromosomal microarray plus Fragile X testing, which is what we would recommend as the first level of genetic testing, and then send whole genome sequencing, which most people are jumping to now not because it picks up things outside of the exome, but it covers the exome better than whole exome sequencing, paradoxically. But if you send both of those, you identify something somewhere between 15 and 20% of the time. So it's not the 4%. I just gave you the most common examples. So, maybe 10% of that are things that are relatively common within that population, by which I mean it's at least 0.1%. And then the remaining 10% or so are things that are incredibly rare, meaning things that have maybe been seen in less than 100 people in the world ever. And so I will say that those precision medicine approaches are still a small number individually, because none of those things individually is more than 1% or so of the autism population, and most of them are much rarer than that. So you pick up meaningful percentages, something like Fragile X syndrome, maybe 1%. That's enough probably for companies to develop a medication, but you get down to 0.1% or below, and it probably is hard for a company to carry that forward. Although we've had examples of single individuals who have a genetic-based intervention developed just for them. So that may actually happen in the future. And then the question is, how meaningful is it for a family to receive that information about their child? And so I think there's the question of if their child already has a diagnosis of autism and we send genetic testing. And I will say I'm always surprised at how impactful it is, even though it may not change management at all. There are a lot of families that still walk around and maybe don't give voice to, well, it was that glass of wine I had before I knew I was pregnant, or it was the stress I was under because we had a rough spot in our marriage, or whatever else. And the genetic diagnosis can be pretty meaningful. It can also be meaningful for family planning, and can be incredibly meaningful for prognosis. So if a child receives a genetic diagnosis that gives them a family group that they can join and see the full range of outcomes for those kids, the family can have in mind what it is that they should think about in terms of goals, and that shapes your intervention target quite a lot. In the case that you get the genetic diagnosis first, right? So the kids who are participating in Guardian, there are some of those genetic diagnoses that are included, like MECP2 mutations that cause Rett syndrome, where that actually can be information that's pretty dramatic to receive early and can give you a sense of what may be coming. And in Rett syndrome, there's actually, and I don't include Rett syndrome here because I don't think of Rett syndrome as being part of the autism spectrum, because by the time those girls are 10, their social skills tend to be an area of relative strength in comparison to motor difficulty and language difficulty, et cetera. So, but if you identify an MECP2 mutation before the child has any sign of Rett syndrome, we now actually have an FDA-approved intervention in Rett syndrome. It's not approved down to 4 months of age, but where you might think about an intervention, and then there are clinical trials that you might participate in. But in a lot of these cases, you're not gonna have that available. And that's part of why we're doing the study, to understand, well, is this information still useful? So we think it's probably useful for epilepsy surveillance, right? But we also think it's likely to be useful for early intervention. So today, so I had I don't know how you describe it, first cousin once removed, my mom's cousin's sibling, so I guess it's my mom's cousin, sorry, had a diagnosis of Down syndrome at birth, was taken immediately away from the family, and ended up dying before the age of 2. That's pretty uncommon in Down syndrome right now. The outcomes in Down syndrome have improved dramatically, and nobody has done a randomized controlled trial to ask whether we should be doing early intervention in Down syndrome. But you see the range of outcomes in Down syndrome is so much better than they used to be. Those kids get access to early intervention as soon as you have the diagnosis of Down syndrome. So that would probably be a meaningful thing to deliver to families as soon as they have a genetic diagnosis that indicates their child will have a neurodevelopmental disorder. And we're not doing it right now, but could we do it? And could that lead to better outcomes? I think so. I don't know how we'll approach that from a clinical trial perspective. I think that's an open question. We're not there yet, but I do think that early intervention is meaningful, even if you don't have something you're going to infuse in the spinal cord.
Yeah, given how heterogeneous autism spectrum disorders are, is there talks or benefit in like creating subcategories or splitting it up? Because
Kathy, you want to?
it's like, I feel like a lot of the treatments, if it's so heterogeneous, it's gonna fall under a statistical significance if we're just treating like 100 different.
Yeah.
Is there talks about it or?
So so the question is whether it would be meaningful, appropriate, useful to at this point have, and I think what you mean is developmental and behaviorally defined subgroups within autism spectrum disorder. And I will say there's been a lot of attention recently to dealing with one of the unintended consequences of the autism spectrum disorder diagnostic category, which is to talk about individuals who have quote unquote profound, there's other words that are used, but profound autism, which unfortunately means that they don't just have autism. I mean, when you say profound autism, it sounds like that means somebody has more severe symptoms of autism. That's not actually what it means in the definition. It means usually that somebody has autism plus intellectual disability and is going to require a substantial level of support throughout their entire lifespan. And by and large, that's defined as that they're going to need constant supervision in one way or another over the course of the lifespan. That may be useful. I'm not sure we have the right words yet. So describing that as profound autism, I'm not sure is the right description. I think it might be a profound neurodevelopmental disorder or something along those lines to really capture that category. I actually don't think we're very good at drawing lines. Kathy has described this as carving meatloaf at the joints. I think, yeah. So I think what we're more likely to see is that it is useful to define the area that is most difficult for the individual in order to define what is an appropriate target of treatment. And we do have the tools actually in the DSM to do that right now, describing the level of support needed in the 2 areas of social communication or repetitive behavior. I think we likely see more examples of that over time. And then I think we can make genetic diagnoses and are likely to see that over time, we may have biomarkers that we can also use to define something more useful. Autism with blank, right? So we have biomarkers like that actually are potentially useful. We just don't have treatments that accompany them at this point. So good question. Yeah, Kathy.
So I have a couple of patients who got genetic testing who are not super low, not profoundly autistic, but not super high, who ended up with these very rare disorders, and then accompanied them to the parent group where there were all these kids in wheelchairs who were really in trouble. I mean, and the geneticists kept saying, this is Stephen Sanders and Matt, that there are probably these milder cases out there that nobody knows. Because I would never predict it from these kids that they would come out positive, but they on anything.
Yes.
but they did. So I just wondered if you think that's true. And then I do think that is for both of the families, it was positive to get this result. But then when I do worry if they got it when the kid was 6 months old, it would have given a very different perspective on what they were, because most of the kids who are identified as having these disorders are so handicapped.
Yeah.
Anyway, but what is that?
No, so so I think the question is about kids who have a genetic diagnosis, but maybe don't fit at least the level of severity the rest of the diagnostic group. So a kid who is more mildly affected. There are examples of that, and I will say we will only really know when we do this prospectively,
Yeah.
right? So we're identifying with, Wendy's screen and Guardian, we're identifying kids who have mutations that you would expect to have an impact. And then we're asking, can we ahead of time predict what that impact is likely to be? And actually what I expect is that we will identify kids who the geneticist would say that kid is going to be in a wheelchair. And then in truth, that they aren't. And why is that? It's because right now we don't do genetic testing in kids who are not as severely affected. And so we're just not finding them. And I think we need to know that information, because if we're thinking about a genetic intervention that has some risk associated with it, or even just pain associated with it, you don't want to intervene at 6 months in the asymptomatic kid who actually may go on to go to college without the intervention. So I do think that there is this challenge. I do think the families like to have the information. I do suspect we're going to hear from some families, and we already hear this, right? Oh, you told me my child would be limited in this way or that way, and they're not. You were wrong. And I think we have to be really careful and humble to say, we don't know the range. We're going to set you up with this parent group, and we'll see for your child how they emerge. Yeah.
Thank you, Jeremy.
Certainly.
Thank you, everyone, for coming.